Terry Hazen, who's a governor's chair professor at the University of Tennessee and has an appointment at Oak Ridge National Laboratory. He's going to be speaking to us this morning about omics. The fantasy is over, the need for multiple lines of evidence. And really, Professor Hazen needs no introduction. He's been long in the forefront of environmental biotechnology, and he's applied a systems biology integrated omics approach to bioenergy, carbon cycling, and of course bioremediation, including some really interesting assessments that he did in the Gulf oil spill. In an already illustrious career spanning 3 national laboratories, Professor Hazen has offered more than 290 scientific publications and holds 5 patents for bioremediation processes, which are used in 35 states and abroad. He's a Department of Energy distinguished scientist and a world-renowned expert in the field, so we're thrilled to have him here with us today. As many of you who have attended before know, the webinar today is being recorded and it will be available on the resource section of the Microbial Insights website within the next week. I'll send out a link to all registrants as soon as it's available. At the end of the morning's webinar, there'll be a question and answer period, so please go ahead and type your questions into the GoToWebinar software, and Dr. Hazen will answer them at the end of the presentation. If you have additional questions, you can also email us at info@microbe.com, and I'll get with Dr. Hazen and get those answers for you. So without any further delay, I'll go ahead and turn it over to Terry.
Thanks a lot, Dora. I've had a long association of more than 20 years with Microbial Insights and with some of the people working there, including Dr. David White and his wife Sandy White. And so it's a pleasure to again show some of the stuff that we're doing. First, let me talk about what is the omics. And we've seen an explosion of these omics technologies over the last 15 years, and it's even more so in the last 5 years. And so typically they can be genomic types analysis with DNA, transcriptomic analysis with RNA, protein analysis or proteomics, and then we can also have metabolomics, glycomics, lipidomics, fluxomics, looking at metabolic fluxes, and then phenomics, basically looking at the base physiology. I'm going to concentrate today, though, on some of the biases that you may encounter and need to worry about with the DNA, RNA, and proteins in particular. But especially the DNA and RNA. So ecogenomics is really attractive because we have all of these different technologies that we can take an environmental sample and look at the population structure, the functional capabilities, and use functional omics, organism and community systems modeling, sequencing, clone libraries, direct sequencing, qPCR reactions, or in other words, particular enzymes and the population's changes to those, and looking even at specific populations and how they imagine. Now, I am going to base most of this on a paper that we published this last year in Current Opinions in Biotechnology, and I'm going to go through specifically some of these things for routes for biases and the types of biases that you might encounter. I'll cover sample collection, then the nucleic acid extraction, and subsequently the different gene-specific approaches and metagenomic approaches, and some of the basic techniques that most people are using, and then the bioinformatics, and finally the conclusions. And then we'll come back to that at the end. So sample collection, there's a number of routes that you can have biases, and our inability, the inability of culture-dependent methods to assess the vast majority of environmental microbes has made culture-independent methods even more important. and removed some of this limitation that we have. There's really a great wealth of information from phylogenetic and functional diversity contained within microbial communities. But there's potential biases in just the sample collection in terms of what filter material you're going to use, the pore size. Are you going to use a pre-filter to get out some of the sediment or this type of thing? The environment itself, its temperature, pH, metals, salinity. Of course, all of these things can affect transport time and storage. And then are you going to immediately freeze it out in the field? And then the sample size itself, number of replicates, how much sample you're going to have to take. A good rule of thumb is to review prior work performed on a similar community in this environment of interest or closely related communities, and particularly pay attention to estimates of biomass, because that may affect how long it will take you to get the sample and how much sample you may need, and if there's particular biases related to that. In some cases, we need 1,000 liters of water to get enough sample to do a complete metagenome. But the wells will only basically only get out in one day 10 liters. Well, that means you're going to have to span over 10 days, and that creates all sorts of issues in terms of water moving into the area, storm events that might occur, and that sort of thing. So you need to be cognizant of all of these potential biases. Also, it'll give you an idea of known community members, and then chemical parameters and physical parameters that you may want to keep track of and show, because I do a lot of reviews for different environmental journals, and I've seen quite a bit over the past few years of people coming in with a single sample with no biogeochemical parameters, other than a particular sequence or a particular array. And if it's that way these days, it'll be almost automatically rejected. We were also getting away from DGGE and T-RIFLIP and things like that because they're low resolution. So I'm going to concentrate on the higher resolution technologies today. So in addition to the sample collection itself, then you have another bias in getting the nucleic acid extraction technique. And you have to worry about whether it's RNA or DNA and that sort of thing. A major concern will be your ability to lyse and recover all the nucleic acids from every microbe in that community. Gram-positive bacteria are notorious for being difficult to lyse, and you need to confirm that the extraction protocol that you're using is tested on cultured strains before using that method on environmental samples. And that means perhaps a mix, and don't assume that these different environments that haven't been tested will have the same properties that something that's been published that's completely unrelated. Commercially available kits are like Powerlyzer and Power Soil, Power Water, and a bunch of things like this, and bead milling approaches. These approaches rely upon a combination of physical and chemical processes to lyse the cells. And one concern, for example, with bead beading is that there's the possibility of shearing the nucleic acids, thus lowering the overall quantity and quality of the intact DNA and RNA in that sample. Now, let me show you some tests that we did in our laboratory with extraction methods. So we basically took a gram-positive, a Streptococcus strain, and a gram-negative, Elchinovorax, especially since we've been looking at oil biodegradation, and take 1 ml culture, diluted it, and put it into some sterile culture media, and then 10 milliliters of diluted culture was filtered onto a 47 millimeter filter membrane, either nylon or PES. These filters were used in either the Mobile Rapid Water Kit or on using the modified Miller extraction protocol, which is in our paper in Science in the supplemental, which we published in 2010. Now, as you can see here, the Mobile Rapid Water and the Miller SDS method, and this is for split samples, they're wildly different on the DNA concentration in terms of almost orders of magnitude difference in terms of nanograms per microliter, and DNA yield in terms of micrograms per filter. So these techniques can be very, very different. Now, that doesn't say anything about the quality, so you also need to look and see what that DNA looks like. And of course, here you can see that we have, with the Miller technique, we have a large amount of DNA, and we can see clearly there's not too much in the way of, there's not too much in the way of degradation and shearing that may have gone on. Because the yields are so much higher, that is something that we decided we would use for this particular study that we were doing. We would use the Miller technique. Now, we've also took these individual species and done different types of filters and looked at the DNA yield. And you can see here they are pretty different too. In some cases, quite a bit different. And we've got pure DNA and looking at how we can recover even pure DNA. And you can see that we get greater than 50% recovered from those filters. We are able to recover DNA from gram-negative and gram-positives using PES filters. The Steravix and some other things are a little bit less, and so we opted to use the PES filters in this case. Now, we've also looked at, in the Mobile, we said rapid water, we tried to optimize it in terms of the amount of time we spent in basically vortexing it, so low speed for 5 minutes or low speed for continuously, and you can see the yields are quite a bit different for these different gram negatives and gram positives depending on what we did. The gram negatives are a little bit closer together, but you can see that we got a lot more out of the high speed for 5 minutes with the gram positive, a lot more DNA per milliliter of culture. And also looking at this, we can look at the quality of that DNA under low speed, medium speed, and high speed, and see if it's being sheared and that sort of thing. These are sort of basic things that before you enter into doing a bunch of sampling, environmental sampling, that you need to consider very carefully. Now, let's move on to the amplification techniques and then also both gene-specific approaches and metagenomic approaches. So a lot of people use the multiple displacement amplification technique, which we published in 2006. And it's for all samples that are low in quantity, potentially have low amounts of DNA and low amounts of biomass. People have been using this, and I'm actually surprised, even though we did this work, and it was actually done, the original extractions were done in my laboratory at Berkeley Lab, and looking at we did some mixes and also some samples from the environment from Oak Ridge National Laboratory. And you can see the number of sequences that we had pre-MDA here, and in this column, and the percentage of those sequences there. Oops. And then you can also see here when we did 1 to 100 dilution, and then 1 to 10,000 dilution. Now, look at, in particular, look at Shewanella. So you see that we had 19% here of the Shewanella in the pre-MDA, but it was 65% of the sequences when we represented 65% of the sequences when we did just a 1 to 100 dilution. When we did a 1 to 10,000 dilution, it dropped back a little bit to 53.5, but you can see these things change, and there may be some specific biases depending on which amplification technique you're using, and this is a rolling block design. And you can go to this paper here that I listed at the end and see all the details of that, and the supplemental has quite a bit of examples too. Now, so I hope this shows you that there's potentially quite a bit of biases in some of these amplification techniques, and some of them that people just automatically use. I see the MDA technique referenced quite often in papers, and they haven't considered that there may be some biases for the particular environment that they're using it on. So in general, and this is in our paper in Current Opinions, we have sequence-based, gene-specific technologies. And advantages of those are ability to use sequence data to more finely define phylogeny, and two, the ability to identify novel species. Disadvantages are there's biases introduced by the PCR amplification that you use, as we just saw. There's biases introduced with selection of primer pair, and there will be skewed towards dominant organisms because it's a picking technique, so it has that sort of bias that you'd have to take an infinite number of samples in order to detect everything that's there. The file chip is a little bit different because you can immediately analyze a sample without amplification, and you can detect very low abundance organisms. It also may have biases that might be introduced by PCR amplification, and it may have biases introduced with the selection of a primer pair. It also, of course, will only see the organisms where there are probes on that array, and I'll talk a little bit more in detail about that in a minute. Metagenomically, in using sequence-based technologies, the sequence data could be used for bioprospecting. It certainly could be an advantage. We can identify novel gene families, and so just forget about the OTUs, which in some cases, and OTUs I mean operational taxonomic units, or what some people might call species. Species identification for archaea or bacteria is pretty difficult to define these days. And we can assemble these into larger contigs that could be used to link to the taxonomy and to the function in that community. Disadvantages are low abundance organisms, as I said, will not be representative, and their sequences will not be represented. And it's highly dependent upon the depth of sequencing that you're using. So in other words, which particular technology you're going to use and how large those particular number of base pairs that you're going to get in your analysis. And it's limited by the accuracy of the annotation techniques, which we'll talk about later. The geochip, which is a functional gene array, and we'll talk more about that in detail in a minute, we have confidence in the sign function. We're able to detect low abundance genes and low abundance organisms. But it's limited by the functions that are probed for on the chip. It's also no indication of similarity to other known genes, because you potentially have tried to see all the conserved genes that have a particular function, but who knows, in different environments, especially in extreme environments, some of those genes may not have ever been sequenced before and may not be on the array. You're unable to identify novel classes of genes if they are not, if in fact there are novel, especially in extreme environments. Now, the Berkeley phylo chip, which is an Affymetrix array that we've used for the past 15 years or so, in its first, in one of the last versions, it was the G2 phylo chip. It had 500,000 probes, 30,000 sequences. It was not hierarchical, and it didn't have any strain-specific probe sets. And it would identify approximately 9,000 operational taxonomic units. But the G3 phylo chip will identify 1.1 million. It has 1.1 million probes. It has 320,000 sequences. It'll identify 50,000 plus, including archaea and microalgae and other things. It's also hierarchical. So if you do not detect a particular probe, there will be a probe that detects at a higher level. So if you don't detect at the strain level, genus level, and then subsequently subfamilies and families, etc., there will be probes that hierarchically will detect those. So even if you can't detect a specific OTU, you can detect that there was a particular family or subfamily there. It's fast, it's reliable. You can basically do these tests on these arrays in about 8 hours. It'll detect pathogens. You can use them for fingerprints, for contamination, for source tracking. We've used them for water, soil, crops, food, blood, guts, skin. This is technology that was developed by Berkeley Lab, in particular Gary Anderson, and it will detect bacteria that will not grow too. And because it doesn't rely upon sort of the sampling techniques that the sequencing and clone libraries do, it's much better at detecting very low concentrations. So the relative, and of course it's very small, the relative location intensity of the fluorescence determines the occurrence and relative abundance of particular 16S rRNA genes, and they're using a 25 mer or 25 base pair. And it also has checking on it for basically perfect and imperfect matches. And so it's highly redundant, and this helps in testing a lot of different things. You can even look at the ratio of perfect to imperfect matches that are made. And if you're getting a lot of imperfect matches, it might suggest that there's some novel species in that particular environment. Though with 50,000 OTUs, it usually detects most of what's there. Now, we published a paper on PCR amplification with the phylo chip for this high-density microarray. And here you can see we were using PCR DNA with secondary sewage, and then we were also doing double-stranded cDNA from our RNA. So this was a direct analysis. And using this non-metric dimensional scaling analysis, which basically shows the entire microbial community as a single spot. And so these are different samples. And it shows how they cluster together. You can see here that you can get pretty different results depending on whether you did PCR amplification prior to that or just did double-stranded cDNA analysis. So especially here you see in the secondary sewage, it's quite a bit different. And with the cDNA, it's double-stranded cDNA, all of them are very close together. But there's a big difference also here with the tropical forest soil, but not so much with chromium-contaminated groundwater. Those are pretty close together. So you can see all the details in this paper. But again, suggesting you need to think about how exactly you're going to analyze the particular samples that you have and how important this is going to be. Now, GeoChip is a high throughput metagenomic technique, and just talking about metagenomic techniques, they're basically, we can have open format detection, and this is from Joe Zhao's lab at University of Oklahoma, and he's the developer of the GeoChip. But using open formats, we could not assure the same genes, proteins, and organs can be compared across different samples. The results could be expected and thus are open. High throughput sequencing, we'll show some comparison of that in a minute. And closed format would be the biolog, the phylochip, or the GeoChip, which are specific arrays. They could be towards particular genes or particular proteins and find particular OTUs. And of course, the biolog plates can do basically phenotypic typing. Now, the GeoChip has gone through a lot of modifications. You can see in 2002, there were 2,400 genes. In 2005, there's 24,000 probes and 150 gene families. And then 28,000 probes and 292 gene families for GeoChip 3, 83,000 probes. And then for GeoChip 5, which is 2013, we have 268,000 probes and 2,433 gene families. Well, if you did your analysis using GeoChip 4 or GeoChip 3, you may not have the same results at all if you now did it on GeoChip 5. And the number of probes has increased because we find more and more, for example, for particular NIF genes. We find a large number, we found a large number of NIF genes, or nitrogen fixation genes, that were unrecognized before. And so every time we go through a modification of the GeoChip, basically Joe has added and his team has added more and more probes to make sure they can detect everything that's there. Now, let's look at the GeoChip in terms of its analysis. You do a DNA extraction from environmental sample. You do whole community amplification. You can use as little as 1 to 10 nanograms of DNA. But you can also do direct labeling. And then the DNA is labeled with Cy5. You hybridize it in the GeoChip at 67 degrees with 10% formamide. And then you do the data processing and statistical analysis. Now, there's a number of different potential errors here, and exactly how the DNA is labeled. And this goes for the phylochip too, and how it's labeled, how well your laser is functioning in terms of determining the fluorescence, and your detection machine is working. So there's a number of different things that can potentially change your analysis. Now, some theoretical predictions. You have a microbial community of 50 taxa, 5,000 individuals, and you use open format versus closed format. You can see even with the sequencing, we get quite a bit of difference. So the similarity is in two samples is quite a bit different. So this is something that you clearly need to be aware of. You get a little bit less of that, 92% and 95% similarity with the GeoChip. Phylochip gives similar results with a split sample. So those can be quite similar. If we look at the GeoChip versus a 16S amplicon versus shotgun metagenome sequencing, you can see with a control and a warming experiment, and this is a paper that Joe's published on climate change and in warming of soil. You can see that with the GeoChip, they get some nice clustering with the control versus the warming. It's difficult to decipher anything from the 16S or the shotgun. Now, we can do all sorts of arguments about which one is best. Of course, Joe says the GeoChip is best, okay? But this just shows that you need to look at some of these things very, very carefully. Now, the paper that was published here recently in 2013 by Mircea Podar's group at Oak Ridge National Laboratory. He looked at 454 and Illumina sequencing for archaea and bacteria and looked at the percent of the community and basically this is an observed divided by expected. So if it was in complete agreement, it would be 1. So we can see as the samples, the percentage of the community for a particular archaea bacteria gets smaller and smaller, the amount of error that you're going to have gets larger and larger. Now, it's not so much if you have, if it's a very large component of the community, say 10%, but once you get down below 1% of the community, you have to worry about the accuracy of what you're seeing. From his paper also, you can see using IMG-M and MG-RAST with the 454 and MG-RAST with Illumina, looking at different major groups of bacteria, you get differences. So the Crinarchaeota and the Euarchaeota are overestimated, observed divided by expected, but the Nanarchaeota are underestimated. And so, and you see differences for both the 454 and the Illumina too, all along the line. So again, suggesting you need to have some multiple lines of evidence to make your final decisions on your conclusions. So once we've done all this analysis, of course, we're doing the bioinformatics and looking at this very carefully, and these can get quite complicated. So in doing the bioinformatics, we're doing sequence equality checking, we're doing OTU table generation with high quality, with FASTQ, FASTA, and this just shows some typical pipelines for doing that analysis. And of course, different programs have different biases and can give you different results. So recently, in Eric Alm's lab, Dr. Sarah Preheim, and she graciously gave me these slides yesterday to show to you, um, did a comparison using data that we had from our Enigma project. And this is data from Dwayne Elias's samples that were taken from above ground reactors that we have at Oak Ridge. And they basically have three different analysis pipelines from three different institutions. So they have the Oak Ridge, University of Oklahoma, labeled OU. We have the Oak Ridge, which is labeled UPARS Chyme. And then the MIT, which is labeled DBC. So we did comparisons between all of these. So the really interesting thing is there's a high correlation between different pipelines at the sequence level. So the sequences are true, but when we do the OTU sort of identification and go down to the genus level, you can see this is a comparison of the Oak Ridge with the MIT counts. And there's quite a bit of variation here. We also see the same thing with the Oak Ridge and the Oklahoma counts, identifications from the OTUs. So actually, your identifications are slightly different. And in some cases may be greatly different for different OTUs. So depending on which software you use, you may get completely different results. And here we have the Oklahoma versus MIT. So again, all three gave slightly different results. So don't get rid of your sequences, because you may have to go back to that at some point and see what it actually is now according to the latest software that's available. So just to summarize all this, you have to look, and all of these things are multiplicative as you move down from A to E, and you see that you have to really judge whether your biases are too great. Are the conclusions correct? Did you end up with the same results using completely different techniques? So that suggests that you have the right conclusions. There's no getting around the fact that multiple, basically multiple lines of evidence are critical. Now here's a particular example that we used on Deepwater Horizon in using the systems biology approach. And we were using physical-chemical analyses. We were using phospholipid fatty acid analysis, metatranscriptomics, isolation and phenotypic characterization and cell counts, cloning and sequencing, 454, phylochip, geochip, metagenomics, and single-cell sequencing, and synchrotron imaging. Now, to get that paper in Science, I knew it was going to be difficult, and I needed multiple lines of evidence. So we decided right from the very beginning that we needed to have all of these lines of evidence, and this is why we got that paper out so fast, because we did this right from the very beginning and had already done all of the background checking on particular techniques. So systems biology, the whole is greater than the sum of its parts, and you really need multiple lines of evidence. And these are some of the multiple lines of evidence that you can have. And this comes from another paper that we have in Current Opinions in Biotechnology. You have ecosystem level measurements that you can take. And you have community level measurements that you can take in terms of metagenomics, 16S RNA clone libraries, etc. You have population level methods that you can use. You have cell level with isolation, enzyme activity, fluxomics, and fluorescence in situ hybridization. You also have protein, lipid, and metabolites for protein, lipid identification and metabolite profiling. And I always like to use particularly PLFA because the PLFA is looking at lipids and particular signature compounds that might be found in the cell wall or cell membrane. And that's a completely different technique than looking at the RNA and DNA. So I like to compare those two and make sure that they're giving me the same conclusions about what's going on. And then, of course, nucleic acids and whole genome sequencing and transcriptomics. A lot of this work that I showed you is part of our Enigma program, Ecosystems and Networks Integrated with Genes and Molecular Assemblies. This is funded by the U.S. Department of Energy. Adam Arkin and I started this back 13 years ago. We currently have 150 researchers in this, where our emphasis is on large-scale collaborative science. We're trying to understand microbes, their communities, and their environment, and how these microbes respond to changes in the environment, how they change the environment. And these are all the different institutions. It would take me too long to list all the people, but I'm clearly standing on the shoulders of hundreds of people. Thank you very much.
All right, thank you very much, Dr. Hazen, for the presentation today. So if you have questions, go ahead and type them into the GoToWebinar software, and we'll get Terry to go ahead and answer those for us. Our first question that we have, Terry, is talking about next-generation sequencing, and you can get a lot of results from this type of metagenomic analysis. So do you have recommendations, because it is such a large amount of data that you get, that how you can really drill down to get a better understanding for site characterization?
Let's see if, well, you know, sequencing has come down so much in terms of cost, in terms of speed. I think everybody's going to have a something like the Lumina MySeq in their lab. I have one in my lab, and you can do metagenome sequencing on this, but you know, one kit you can take care of maybe 6 or less metagenome samples, but they're short reads. Longer reads, of course, are better because we can get more information out of those, and potentially we can reconstruct even the enzyme out of those longer reads. So there needs to be a balance between metagenomes and 16S. We've done some work, as you've probably seen me present in a couple of places in Eric Home. We've done some work on their 100-well survey at Oak Ridge, and we can actually predict certain water quality parameters from the 16S microbial community structure. And we can verify that using a number of different techniques and a number of different software. But you're going to have to pick your software very carefully, and you may want to run 2 or 3 different types of software if you're just going to identify OTUs from 16S sequences. Does that answer your question?
Yes. All right. The next question is, for an educated layman, what's the difference that you were talking about between the deep sequencing and other sequencing technologies that you listed in the metagenomic flowchart?
The deep sequencing is basically how long the reads are, and then if you can basically sequence through a large group and keep resequencing it until you reduce the number of what we call contigs. So these are particular sections that we know are grouped together. So if you have 50 contigs for a particular sequence, that's not very good. But if you can get that down to 1 contig or 2 contigs, then potentially you've done enough deep sequencing to understand exactly everything that's potentially in that organism.
So you mentioned having the MySeq in your lab. Do you find that the length of the sequence is sufficient for what you were just talking about with the MySeq, or do you need some of the 454 with longer sequences as well?
Well, the 454 is rapidly falling out of use. In fact, we traded our 454 in on a MySeq um, with Illumina. We and we have a high-seq, but because of the cost, a lot of people are are electing to use the shorter reads that are on the MySeq. A kit on the MySeq costs several $100s, maybe as much as $900. But from that, you can do 90 16S sequences, samples, I should say. And you can do maybe up to 6 metagenomes. So my MySeq is being run all the time. And basically, it may take a day to do a complete run, or 24 or slightly longer. But of course, then you have to do the analysis and figure out exactly how you're going to identify those OTUs, or what program you're going to use to identify those OTUs.
Okay. So do you feel like, since there's uncertainties that are still left after we do the omics analysis, do you think that the statistical portion is becoming more and more important in what we're doing?
Yeah, and doing repeated samplings and having replicates and doing very careful planning. And so I saw a great bird's eye view lecture by Dr. Jim Prosser at the International Symposium on Microbial Ecology in South Korea 2 weeks ago. And he didn't have a single slide, but his point was well taken, that he did some analysis of environmental microbiology journals over the past several months and looked at all the papers in them. And 43% of them, he did not see any question being asked or any hypotheses being tested. And that's still a very important thing for us to try to emphasize. Don't just do descriptive stuff. And a lot of people are enamored with getting a particular sequence and publishing it right away. And these genome announcements are for that. And I put in genome announcements and had them come out in 2 weeks from the time I put them in. So these can be very fast, but they're very short. And it basically just tells people that you have done a particular sequence and they can find it here at your particular site where you've archived it. But more importantly, we need to do more of this planning and designing experiments that test hypotheses and answer questions, okay? And showing experimental setup and the techniques that are going to be used, with all of the caveats that I showed you about what needs to be done in the background to sort of control and prove that. This is getting to be more and more critical. And as reviewers, if you just do one particular technique at one particular site, we don't see any other biogeochemistry or anything else describing that site, we're going to reject it automatically. We're not even going to review it. We're going to scan through it and send it back to the editor and saying, reject this. So, there's got to be more proof in what you're doing, and you have to have better quality data. The omics is quite fascinating because of all the things it can potentially show you. But it's got to be married to good experimental design.
That makes
Sorry,
sense.
that was a long answer.
No, that was a good. Okay, so can any of the methods that you've talked about today be used to identify mutations that may occur in the microbes or in communities resulting from a change in the subsurface, such as an introduction of a new contaminant?
Yeah, of course, metagenomes can, and you could also do isolation. We're doing this too. We're isolating deep water and deep sediment samples from around the world right now as part of a project that I have with BP in looking at deep water sites where they're going to potentially be drilling. And we see wildly different abilities for some of these microbes to degrade oil. And so we're scratching our heads about that because the 16S identification would say that they're 99.99% similar. So what we're doing now is doing deep sequencing on those individual isolates and trying to see if there's just minor differences in some of the like PAH degrading enzymes genes. Because maybe only, a minor change could result in a major change in the ability to degrade a particular contaminant, for example, or a particular oil or a particular hydrocarbon. So that is one thing that we need to do more and more, is to do some isolations and do some deep sequencing on things that we think are absolutely the same, even though when we look at them phenotypically, they're different.
Okay. So given the number of biases that you've talked about today, do you think the application of these microbiological tools can be complicated, like in court cases, or cause problems?
Oh, yeah. So it could be, and in fact, there is a group that has been training judges, and I was involved with this for several years, and we went out to training circuit court judges and state judges and even Supreme Court, state Supreme Court judges, and we had one Supreme Court justice come one time, and internationally too. And we basically go through some mock scenarios and try to, of course, they always consider that everything's black and white. And, science in some of these areas is not black and white, it's gray. As you can see, depending on which techniques you used and do you have enough information to show exactly, how well that particular technique was worked, and could you use something else that could verify that in a completely different avenue. So we have done training of them, and so there are biotechnology expert judges now. And of course, a lot of lawyers came to these sessions that we had. And this is called the ASTAR program. So if you look up ASTAR, you can find where they've done this particular type of training. So yes, even the judges recognize that it's very important. I also, of course, stress that they're expert witnesses. Quite often they want to do due diligence on their expert witnesses and look if they publish papers in those particular areas and if they really have expertise in those areas. And I even showed them how to look up their H-index and do Google searches and do Web of Science searches on their papers and that sort of thing. And so they were very interested in having that ability to look at supposed expert witnesses.
I guess, do you think with this training and with the fact that these tools are becoming used more frequently, that this is going to change and that there will be experts that are be available soon?
Well, there's always experts available. Um, you know, a lot of people have set up consulting companies just to do expert witness testimony. So, and there are some that are clearly very well qualified to do that. Though quite often, um, there are folks that have gotten out of research and are not doing as much um, as they should be to keep up with the state of the science.
Okay. The next question is, I haven't seen the article on the Deep Horizon incident that Dora mentioned in the beginning of your talk. Can you summarize the take-home message of that article?
Yeah, so the take-home message was it came out in August of 2010 on Eureka Alerts, and basically it described that there was a microbial community in the deep, and this is at 1,400 meters, with a because there was an active plume at 1,100 meters that was coming out from the oil in the deep. And we showed that there were bacteria down there that were degrading that. Now, this is, um, a lot of people thought pressure would be an issue. It wasn't. We showed that. And they also thought temperature would be an issue because, of course, once you get down below 400 meters in the Gulf of Mexico, the temperature is 4 degrees all the way to the bottom, maximum density of water. Well, that was not the case either. In fact, we showed that the half-life using 4 different methods for the bacteria in that deep water plume, for the oil in that deep water plume, was 1.3 to 6.1 days. Now, of course, that doesn't mean all the oil was degraded in that time, because just like half-life for oil, every time you degrade half of it's left, because there are components that degrade more slowly. But we showed it completely disappeared. In fact, it represent. So the well was capped in July 15th. By August, the first week in August, we could not find any oil in the deep, and it's because of this dispersion and biodegradation. When that paper came out, of course, it explained why they couldn't, NOAA and everybody else couldn't find any oil pretty rapidly after the well was capped, the Macondo well was capped. I gave over 500 interviews and lost count. If you type in my name into any web browser, you will find a lot of those interviews, thousands and thousands of them. I still give about 1 interview a week on average. So that Deepwater Horizon event is going to last for a long time. But this is what I had in terms of, I had multiple lines of evidence. We published about 20 papers on this. We'll probably publish about 20 more. Over 10,000 samples were taken during the response phase. So we have a tremendous data set. And then subsequently, I'm looking at other deep water sites around the world where potentially they're going to be doing deep water drilling.
Yeah, I like the pictures of the biotrap samplers that were deployed at the depths there. That was a really interesting work with the biotraps.
And of course, those came from Microbial Insights. And so we have a paper that should be coming out shortly on that. And we were able to string these bug traps with Macondo oil on them and some without. And because these bug traps, and there's little 3 millimeter diameter beads, and because they bacteria will rapidly colonize those. And of course, we call them bug traps because they're sort of baited. So in fact, we see that in fact they have a number of of biofilms and that sort of thing. So basically, it's very easy for us to extract both lipids and DNA and RNA and a variety of other things off of these little 3 millimeter beads. And they're, sorry about that, they're basically um, showed that in the different depths that we put them at on the um, riser in the, this is one of the relief wells next to the Macondo well. We put them all the way down to 14 and 1500 meters. At every level, it was a different microbial community that was degrading the oil. So we could show that by the phylochip with 16S. We've done some other sequencing on that too. And that paper should be coming out shortly.
That's great. All right, well, thank you very much, Dr. Hazen, for your presentation today, and thank you, everyone, for joining us. We'll have this available on our website for anyone that would like to go back and revisit it. And Dr. Hazen did say he would make a PDF available of the presentation. If anyone's interested, you can email us, and we'll be glad to send that over to you. But thank you again, Terry, for your time today. We really appreciate it.
Thank you. And remember, for all of you, I'm only a cybersecond away. You can get me at tchazen at utk.edu.
All right, thank you. Have a good day. Thanks.